Assalamu alaikum and hello to everyone. This is Bajem here, and this is my video on the 16th chapter of A2 Biology, which is genetic inheritance. Remember that this chapter we also learned about it in the AS, which was the previous part of this chapter, which is about mitosis and asexual reproduction. This chapter is the continuation of the same chapter, but this consists of sexual reproduction. Don't worry, we're gonna learn about it. Pretty simple topic. Let's get into it. Okay, so before hopping into the chapter, let's learn about the simple terms that are gonna be used in this chapter. Okay, so gene, allele, trait. These three terms they're gonna be used frequently in this chapter. So remember to memorize their definition correctly. I mean, when I was a student, I used to struggle with these definitions a lot. I mean, I used to mix up genotype, I used to mix up gene, I used to mix up uh, chromatid, I used to mix up everything. So, chromosome, chromatid, I used to think it's the same thing. So I just I just used to call a gene a chromatid 2 and a gene a chromosome 2. So please do not mix them up. Make your concepts clear. Learn them properly. What is a gene? The basic molecule is a DNA. A DNA is we learn about the DNA, deoxyribonucleic acid, which contains the genetic information of our body. So, what is a gene? A gene is a length of DNA that codes for a specific amino acid. A length of DNA that codes for a specific amino acid is called a gene. And in the length, a base sequence of DNA, it's written here, but I'm just telling you the most easiest definition that you guys need to memorize. It is the length of DNA that codes for a specific amino acid. It's called a gene. Allele, what is an allele? An allele is a variant form of a gene or a variety of a gene. For example, if there's a gene that's coding for hairs, right? Hair color. Let's take an example of hair color. So there's a gene that's coding for hair color, and the color on the gene is black. Now there's gonna be another variety of the same gene, the same color. There's gonna be another color for it. For example, there can be another color which is brown for hair color. So the gene is black because it's being transferred from parents. So the gene is going to be black for it. The main color that is being expressed is black. But the allele, the variety of the gene, another form of the hair color, another type of hair color is gonna be um, brown. So remember always that the difference between gene and allele is basically that an allele is a variety of a gene. You can say that allele is also a gene, but it's a variety of a gene. The main is not the allele. The main is the gene. Okay, but if there's another variety of it, it's gonna be an allele. So always remember what is an allele. Allele is a variant form of a gene. What is a gene? A length of DNA that codes for a specific amino acid. A trait here, pretty simple. Any characteristic of an organism can be skin color, can be hair color, can be height, can be anything. So a trait is basically a characteristic of our quality, the tendency of someone or something. So these were some basic definitions. Now let's hop into the main, the main journey of the chapter. Okay, so genetic inheritance is the process of transferring traits from parents to offspring. Traits we just learned characteristics. So we can say that genetic inheritance is the process of transferring characteristics from parents to offspring. Remember that we have things common with our parents. These characteristics are therefore transferred from parents to offspring by the process of genetic inheritance during both sexual as well as asexual reproduction. We learned about asexual reproduction before, which happened due to mitosis, which was because of the exact same copies of the cells of the gametes. You know, like the what do you call it? The zygote, but that doesn't happen in humans. I mean, not as in, reproduction for an embryo. So we're not gonna. I mean, we finished that chapter. Now we're gonna learn about meiosis. Meiosis is what happens in the mammals. What happens, you know, what causes variation in the offspring and the parents. Because we're not exactly the same as our parents. Of course, we're different from them. There's a difference between we can differentiate between parent and offspring. So there's a variation between them, right? And this variation occurs because of meiosis. Let's see that how. It is done through passing the genetic material, DNA, from parents to offspring. Remember that you don't need to transfer anything else except for the DNA. The DNA is the information of the body of the parents, right? So if that information transfers into the offspring, of course, they are going to get the information that there are going to be some characteristics that are in common with the parents. So remember that how does it happen? It happens by the transferring of DNA from parents to offspring. Now we're going to learn about meiosis. Meiosis is a simple cell division process that gives rise to genetic variation. I just told you what is genetic variation. Variation in parents and offspring. Its main role is to produce haploid cells from diploid cells. Now I'm going to tell you what is the difference between haploid and diploid cells. So this is right here. You can see the one on the left is the haploid cell. What is the difference between haploid and diploid? In a haploid cell, there is not a complete set of DNA, of chromosome. You guys can see it doesn't have a sister chromatid. The green one doesn't have a sister chromatid. Whereas in the diploid cell, you guys can see that they have sister chromatids. There's a complete set of chromosomes. For example, if there was going to be three chromosomes here, so there were also going to be six chromosomes in the homologous. No, sorry, not the homologous, the diploid cell. But in the haploid cell, there are always going to be three, so it's an incomplete cell, right? It doesn't have a sister chromatid with it. So whereas in the diploid cell, you guys can see that there's the whole set of chromosome. So it's a complete set of chromosome. This is the main difference between haploid cells and diploid cells. Haploid cells do not have complete sets of chromosome. Diploid cells have complete sets of chromosome. Okay, now let's get into the meiosis. What is meiosis? Meiosis is we learned that it's a process of cell division which causes genetic variation in the parents and offspring. So there are always even in mitosis we learned that there are four processes: PMAT, prophase, metaphase, anaphase, telophase. Even in here, remember four processes. Learn the term PMAT, prophase, metaphase, anaphase, and telophase. But the difference is that here in meiosis this occurs two times and it's different both times. It's not the same thing happening two times. It's different the both times. So prophase one, metaphase one, anaphase one, telophase one, then prophase two, metaphase two, anaphase two, telophase two. This is completely different, okay? And the first step taking place here is a diploid cell. And in a diploid cell, there is a complete set of chromosomes. In a haploid cell, which is the result of meiosis, is not a complete set of chromosome. It has an incomplete set of chromosome. So let's get into meiosis. Um, we start from a diploid cell. Uh, so let's get into after interphase G1. What is going on? Interphase S1. Sorry, S. S. We remember that what happens is replication occurs here. Now, what is going on? That the chromosomes they get their centromeres. When they get their centromeres, they arrange like that. You guys can see these centers that are attached to them. Before in the G phase, you can see these centers, these white colors, kinetochores, proteins containing a centromere. So these were absent in the G1, and what happens in the S phase is that these centromeres they appear. Now this is where it enters meiosis prophase 1 after G2. What happens in the prophase? A little bit of spindle fibers start forming. You guys can see that there are these webs. It looks like a spider, spinning its webs. So remember that these gray color things, they're called centrosomes. They are present in the cell. So these centrosomes, what they do is they make spindle fibers. Centrioles. Okay, these are called centrioles. And what do the centrioles do? They make spindle fibers. These spindle fibers are web-like structures. These web-like structures, what happen is they start being created in prophase one. Just remember this. What is the main process occurring in prophase one? The spindle fibers, they start being made by the centrioles, and the nuclear envelope disappears. These are the two main things occurring in prophase one. Remember what? Spindle fiber, nuclear envelope disappears, spindle fiber starts forming. Metaphase one, the cell enters metaphase one. What is happening here? You guys can see clearly that the these spindle fibers, they go and attach to these centromeres of the chromosomes, and these centromeres, they align on each other. You guys can see here these centromeres, they combine with each other here. Basically, they align on the center, but they also like kind of overlap and combine with each other, right? You guys can see here. Now this is a very random process. This is not according to some plan or something. This is a random process that is occurring here. So this therefore causes variation. And this process is called independent assortment. This is called what? This is called independent assortment. Independent assortment is basically the production of different combinations of alleles in daughter cells, a result of random alignment of bivalents on the equator of the spindle during metaphase 1 of meiosis. So what is going on here? Independent assortment, because these chromosomes, they're when the spindle they attach to the centromeres, these chromosomes they align with each other. When they align, they overlap. This overlapping causes combinations of various alleles, various uh, genes, alleles, thus causing variation. Therefore, during this mixing, there's variation. Therefore, now what happens in anaphase one? In anaphase one, these chromosomes, the spindle fibers, uh, they start moving back. They start stretching back towards the opposite ends. What does this happen? Just like in mitosis, when these central chromosomes are pulled back, due to this pulling back, they get separated from each other. After overlapping, they get separated from each other, which causes them to consist of what they overlapped. So they're gonna now stick to the same information that they overlap with, and therefore it became a part of them. And now they're gonna contain of those traits that they equipped, you can say, from each other. Now this mixing occurring in anaphase one, pulling back of spindle causes them to go to the opposite poles. These two are the poles here, the centrioles. So the spindle fiber, when they pull them back, it causes the chromosomes to go to the opposite poles. Therefore, two cells form here. You guys can see two cells forming here, and these two cells form in the process of telophase. Now, how do these two cells form? The nuclear, and when these two they're separated, right? One goes here, one goes here, one goes here, one goes here. Now these two cells are gonna get separated. A nucleus is gonna form. A nuclear membrane is gonna form around them. Around this, two nuclear membranes form. After the formation of nuclear membrane or the nuclear envelope, what is gonna happen? Cell membrane is gonna divide the both cells. So here a cell membrane is dividing the both cells. Therefore, two cells results. So now the part of meiosis 2 is gonna start, where prophase 2, metaphase 2, anaphase 2, and telophase 2 are gonna take place. Now what do we have here is cells with two chromosomes. One cell has two chromosomes, another one is two chromosomes. We started from diploid where we had four sets of chromosomes, but here we have only two. So they split up into two, right? Now prophase 1, what is going on? Again, the same process which is going to be here. Um, nuclear envelope's gonna disappear. Okay, so what is the difference in Prophase 1 and Prophase 2? We have two cells, but in both of them, the nuclear envelope disappears and spindle formation starts, just like in Prophase 1. Now, when spindle formation starts, it's gonna go and attach to the centromeres of the two chromosomes present, and due to this, it's gonna pull the chromatids apart. Now, here, difference in anaphase 1 and anaphase 2 is that here the chromosomes were being pulled apart, and here the chromatids are being pulled apart. This is the difference here. That in anaphase 1 chromosomes were being pulled apart, anaphase 2 the chromatids are being pulled apart. Due to the pulling apart of these chromatids, 1, 2, 3, 4. 1, 2, 3, 4. 2 on one side, 2 on the other side, 2 on one side, 2 on the other side. 4 telophase in telophase 2, 4 cells are formed. 4 nuclear envelopes formed. Uh, 4 new membrane, cell membrane, cell surface membrane is going to separate these cells, and therefore 4 new cells, 4 haploid cells formed there, 4 cells with incomplete sets of chromosomes formed there. So all the way from diploid cells to haploid cells. Okay, remember where independent assortment takes place? Independent assortment takes place in the metaphase one. Okay, I can show you guys a little bit of the structure here. Wait, yep, this one. This is how it actually occurs. So yeah, this is how it starts. You can say prophase one, nuclear envelope disappears. There you go. The central, uh, this is happening here now. Okay, metaphase, anaphase taking place now. Anaphase one, we'll go to the opposite poles, the chromosomes. Nuclear membranes form. 2 cells separate. Telophase one done. Prophase, metaphase now. Metaphase two pulls the chromatids apart. Anaphase the telophase, 4 new membranes formed, separated from each other, 4 haploid cells formed. You're done with it. Okay. Now meiosis produces genetically different cells, and this happens through crossing over of chromatids and independent assortment of chromosomes. We learned about the interdependent assortment, which happens in metaphase one, and the crossing over happens in prophase one. You guys can see here that it crossing over. You see, this crossing over, they combine with each other. You guys can see, right? These mixing up, and the what do you call independent assortment? It takes place in metaphase one. So prophase one, crossing over, metaphase one, independent assortment. Boom. Okay. Now is the time for learning monohybrid and dihybrid causes. Before that, let's get into the simple definitions of heterozygous and homozygous. Uh, Remember that what is a heterozygous? Hetero means different. So heterozygous means you know this zyg nucleus. So heterozygous means different alleles of a particular gene, having 2 different alleles of a particular gene. Remember, in a single gene there are always um, two types of alleles, right? One can be remember that always there are 2 types of gene. One is the dominant and one is the recessive. In one gene there are 2 types of alleles, and these there are also 2 types of allele. One is dominant, one is recessive, and we have learned this before, but I'm just telling you that in one gene there are 2 types of alleles, and in these alleles there's one dominant, one recessive. The dominant allele is always represented by a capital letter. For example, here the R is capital, so the R is going to represent the dominant one. The small r is going to the smaller is going to represent the small one. So in a heterozygous, what happens is that heterozygous cell, it has 2 different alleles of a particular gene. So in one gene there are 2, right? And what happens in the heterozygous is that in one gene there are going to be 2 different types of alleles. For example, there can be one black, one brown. But if the black one's dominant, so only the black one's gonna be expressed. Therefore, okay. And in a homozygous, homo means the same. So in a homozygous, what happens is two identical alleles. For example, RR, it can be either capital R, or a small r, small r. But in heterozygous, it was different, so it's going to be capital R, small r. We always write the capital one first, then the small one. And if it's a heterozygous, if it's identical, then you can write both. So what is the difference? Heterozygous contains two different types of alleles in one gene. Homozygous consists of two similar types of alleles in one gene. Capital R, small r, small r. Homozygous, heterozygous, done. Okay. Let's get here, which is basically, you guys can see there are two types of parrots. One is yellow, one is green. Now, I told you in one gene there are, two different types of, what do you call it, alleles. So we can see here that there is capital G, small g, capital G, small g. Remember that here the dominant one is the capital one, so the dominant one is the green one, right? You guys can see here that the dominant one is the green one, and the small one, small g, is the yellow one, which is the recessive one. Okay, just let me check one thing. Okay, now we're gonna learn here genotype and phenotype. What is the difference between a genotype and a phenotype? I remember I told you guys that I used to confuse them a lot. Genes, genotypes, chromatids, everything, chromosomes. Do not confuse them. Pretty simple. Is a Chromosome is a molecule which has a molecule of DNA which has two chromatids on them joined together to form one whole, complete chromosome. It has four telomeres and one centromere. So always remember this. What is a chromatid? A chromatid is a single strand of DNA, which we have seen before. Only a single strand of DNA is called a chromatid. Like if you divide the chromosome in half, you're gonna get two chromatids. So these two chromatids are basically one chromatid is one. It consists of two telomeres, and yep. After that, we learn gene. What is a gene? A gene is a length of DNA that codes for a specific amino acid. What is an allele? Allele is a variety of a gene. Now we're going to learn about a genotype. It's totally different from that. A genotype is a sum of genes transferred from parents to offspring. Remember that when we receive something from our parents, they come in the form of genotypes. And in genotypes, remember there are different you know genotypes is something that we cannot see by ourselves. Okay, it's inside our body. And then there's this phenotype. What is a phenotype? Set of observable characteristics. Observable means we can see it, right? So resulting from the interaction of its genotype with the environment. So therefore phenotype is something that is expressed in our body. We can see it: skin color, hair color, anything, height. It happens when our genotypes, they combine with the environmental factors. And when the genotype combine with the environmental factors, there's a result which is called phenotype. And phenotype, we can observe it. Genotype, we cannot observe it. Okay, so remember, in one genotype, there we can focus on a single phenotype. For example, if we talk about if we were seeing the example of parrots here. Wait, yep, we can see the parrots here. That what is happening here in the parrots? That we're talking about color here. Okay, so in when we talk about color, when we talk about the genotype of this parrot, for the color, it's gonna be capital G or small g together with each other. Okay. Okay. Now remember heterozygous and homozygous. Uh, so heterozygous, we can see that this is a heterozygous here, right? One is a capital G, one is a small g. Remember that whenever it's a capital G and a small g together, the capital G color is always going to be expressed. So in a heterozygous, always the dominant allele is expressed. What is the dominant allele here? The dominant allele is the capital G, so therefore this parrot is going to be of a green color. What is how is this yellow color going to be expressed? This is a pretty good question, you know. How is the yellow color going to be expressed? It's only going to be expressed when it's going to be a homozygous with a small g and a small g. So whenever it's going to be homozygous recessive, remember that G is the recessive, the small g one is the recessive. So if it's going to be two G's, only then we're going to get a yellow color parrot because it's recessive, right? So if there's a capital G with a recessive one, of course the capital one is going to be preferred. So how we're going to get a yellow colored bird? Therefore, we need to have two small G's. Two small G's will always give us yellow. Other than that, there's no other way we can get a yellow color G bird. There are three ways in which we'll get green birds. Three ways, which is capital G either, or either a capital G and a small g, which is heterozygous. In homozygous, we have two capital G's. In heterozygous, we have a small g and a capital G. In homozygous recessive, we have two small g's. Now, what is going on here? This is a monohybrid cross. A mono in what happens in a cross? We mate two uh, two organisms that have different genotypes or maybe the same genotypes just to see the results of the offsprings. So what are we mating here? We're in this example we're mating a capital G and a small g. So we're mating a heterozygous molecule, not a molecule, a heterozygous organism with another heterozygous organism. Why heterozygous? Because it has a capital G and a small g. Capital G and a small g. So one heterozygous organism with another heterozygous organism. Now, how does this work? So remember that we're gonna make this table. We're gonna write capital G small g, capital G small g. Now we're gonna you can say we can multiply them. Okay. So now we're gonna when we write them together. So we're gonna write this capital G with the small g here. So GG, capital G small g, capital G capital G. Sorry, not the small g, capital G capital G. So two capital G's here. Here, capital G and a small g. So we always write the capital one first. I told you before. So this is a heterozygous organism being formed. So a genotype being formed. So, so a capital G and a small g. Therefore, we get this. A capital G and a small g again. We get this. A small g and a small g here. This is rare. What is the phenotypic ratio? By phenotypic ratio, we mean green ratio yellow, because there are two types of phenotypes here that are being expressed. One is green, one is yellow. So what is the ratio between them? It's 3 ratio 1 because we get 3 green colored and 1 yellow colored. So the ratio between the the yellow and green, the phenotypic ratio here is 3 ratio 1. You guys understand now? The green color is gonna be 3 birds because 3 ratio will be green birds and 1 ratio is always gonna be yellow color. This is how we do a monohybrid cross. Now we're done with this. Okay, let's get into a dihybrid cross. Now, what happens in a dihybrid cross? When in a monohybrid cross, we learn about only one type of gene being involved, right? In a dihybrid cross, it's di, so it's going to be two types of gene being involved at the same time. So here we can see that there are two types of gene. There was only a 1B a small e here last time, and you can say there's only going to be one b and a capital B here last time. But here we're gonna compare two types of gene at the same time. Now, how to create these? How to create this B, and you know. So what is gonna happen here is that we're gonna combine this B, capital B, with a small b, this small b with this small b, and yeah, I'll show you. So let's get here. Capital B describes the dominant allele and produces a black coat. Remember that the capital one is dominant, and it is because it's capital. right? Because the B is capital here, as you can see. The B is capital, so it codes for a dominant allele and produces a black coat. The small one's gonna be always recessive, like yeah. So it produces a chestnut coat. It's gonna be a chestnut color. For eye color, E, um, a dominant allele, the capital one E, so it produces brown. The small one E, so it produces blue eyes. Remember, B capital is gonna be for black coat. Small b gonna be for chestnut coat. Capital E dominant B, brown eyes. Capital small E recessive blue eyes. Okay, so let's see here. Now these are gametes. How are gametes formed? This is the main question. You guys can see these are one, two, three, four, five, six types of gametes here. How are these gametes formed? These are two from one parent, and these are four from one parent. How do these parents gametes form? Remember, in exams, we were always going to be given two types of genotypes. Okay, these are the genotypes here. We're going to cross these two genotypes. How are we going to get these two genotypes? Okay, remember, now I'm going to zoom in. Okay, how we're going to make these genotypes is basically this capital B, it goes and combines with this E. This capital B combines with the E, capital E here. Therefore, we get BE. Another genotype, another gamete that's going to be formed here is going to be a capital B with a small e. It's just like multiplication over here. You guys can see that you can say there's a bracket around here, okay? So first we're going to multiply the capital B with these two terms, then we're going to multiply the small b with these two terms. So we can say that we can say that the capital B is being combined with the small capital E, then the capital B is going to be combined with the small e. So here capital B E and capital B small e here. Now small b with the capital E, but I always told you we write the capital E first. But here in dihybrid cross, remember that it doesn't matter. It doesn't matter here for the gamete formation. It doesn't matter what you write first. The thing that comes first, the alphabet that comes first. If you're writing B first, then always B is gonna come first. The capital is not gonna matter here. So if you're writing B first, B is always gonna come first, just for discipline and making it easier the question. So remember that if you're writing B first, B is gonna come first. If you're writing E first, then E is gonna come first. No matter if B is capital or E is capital. Okay. So capital B, small b, capital E, small b, small e. Done. 4 gametes formed. Small b, capital E, small b, small e. Therefore, these gametes form. Now here, the same process: capital small b, capital E, small b, small e. Only those two are forming here because if you're gonna do it this small b, so it's gonna give us the same. So we can say that overall only these two gametes are formed here. Now, how to cross them? We write one parent on one side, another parent from one side. Now we can see here that when we combine them, um, this B, we're gonna write the B's first, then the E's after them. So now imagine these two B's being combined, these two gametes being combined. So what is going on? What is gonna happen here? Capital B with the small b and the capital E together. Capital B with the small b and then the two E's. You guys can see here. This is gonna combine with this. This is gonna combine with this. One capital B, one small b, one capital E, and one capital E. Therefore, also the phenotype is gonna be a black coat and brown eyes. Remember that the dominant is always expressed, right? So here, what was dominant? The capital B. So it's gonna be black coat, capital B, black coat, and brown eyes because it's two dominant. So there's not gonna be another option. So brown eyes because it's the dominant one. Here, capital B, small b, capital E, small e. So the dominant one is going to be expressed. Now, what's the dominant way here? It's a black coat with brown eyes. Dominant one's expressed. Just like that, we're going to continue with all of these here. And here we're going to get blue eyes. Two times we're going to get blue eyes. Because why? Because the small e is going to combine with the small e here. The capital B black coat because we have a capital B here. But here it's gonna be both recessive, because we got a small b with a small b and a small e with a small e. Therefore, only recessive traits are gonna be expressed, and therefore a chestnut coat, which is recessive with blue eyes, both recessive, is gonna form. Done. Now test crosses. Remember that what is a test cross? Test cross is used to deduce the genotype of an unknown individual if we know the if you know that is expressing a dominant phenotype. So we know that if one some animal is expressing or a dominant phenotype, right? If you know what is a dominant phenotype in animals, if you know that black color is the dominant phenotype, and we know that it's black, but how we're gonna get its genotype? So to tell the genotype, we can tell it by the test crosses. How it's gonna work? If no offspring exhibit the recessive phenotype, then the unknown genotype is homozygous dominant. Now we're gonna see the example here. See, we know the gametes of one organism here, which is homozygous recessive, and we do not know the you know we do not know the uh, genotype of the other organism. So we just know that this organism is expressing a dominant feature in it. So we don't know, it's is it a heterozygous dominant or a homozygous dominant. Now to figure this out, we're gonna do a test cross. So what are we gonna get here? All four types of heterozygous dominant long years heterozygous dominant organisms here as offsprings. If there's a single offspring which has both recessive, which is the homozygous recessive, if you have a single organism which is a homozygous recessive, it means that the organism that is being tested is also heterozygous. Because remember that if there was a small d here, at one point there was going to be small d with a small d, and it will give us short ears. So therefore, what is written here? If no offspring exhibit the recessive phenotype, so then the unknown genotype is homozygous dominant. If no offspring express the recessive phenotype, right? The recessive one is small d. So if there's no small d here, then it means that the unknown organism is therefore a homozygous dominant. But if there's one offspring with a recessive allele, if one offspring exhibits short ears, that means that the unknown gametes of the organism is therefore heterozygous dominant. Now this was all about our test crosses, and thank you so much for coping and listening to my video. Till then, till next time, see you all. Take care. Bye bye.